It was discovered that gene expression patterns of Burmese pythons change extensively when they eat. Their insulin levels will skyrocket and stay high for several days while the meal is digested. Meanwhile, their gut and heart increases in size along with many other changes. This led the researchers to question if our response to insulin is similar to the python and if insulin possibly regulates far more than what we know. Insulin plays a crucial role in many processes, but what happens when the insulin regulation doesn't work correctly? Lack of insulin or its receptors may lead to diabetes. It is known that people with diabetes suffer from a range of symptoms. Chronic glucose elevation in the blood can cause toxicity leading to these symptoms. Researchers think that not all the symptoms can be explained by glucose toxicity, and insulin may play a role instead. Insulin resistance is highly correlated to cognitive decline. We have receptors that play a part in insulin signaling and have a vital role to play in regulation, metabolism, and reproduction. After a meal, the glucose levels in the blood increases rapidly, causing release of insulin from the pancreas. Insulin then promotes absorption of glucose into the muscles and other organs and inhibits its production in the liver, which in turn results in a dramatic decrease of glucose concentration in the blood. It was discovered that cognitive function, learning, and memory formation was enhanced in humans who were given intravenous and intranasal doses of insulin, respectively. A group of scientists from Dundee, led by Professor MacKintosh, decided to carry out experiments on mice to determine how fasting and systemic insulin signaling regulate phosphorylation. Experiments on mice were carried out by elevating their insulin levels using different methods. After elevation of insulin levels, the mice were killed and their organs were harvested. Molecular techniques were then used to look for specific changes in phosphorylation of proteins in different tissues. Insulin caused phosphorylation of many proteins within the organs and fat tissue of the mice. In the brain, the peripheral insulin had the opposite effect. It caused dephosphorylation of some proteins. These included, for example, MARCs and SRGAP3. Both of these proteins play a role in cell shape and migration via controlling the cytoskeleton. Fasting leads to opposite results in the brain, causing higher phosphorylation of MARCs and SRGAP3. Changes in the brain were particularly surprising for one reason. Insulin is unable to pass the blood-brain barrier, so body or peripheral insulin can't be the molecule directly causing the changes. So what causes the dephosphorylation then? A molecule acting as a mediator is a very likely option. This molecule, or signaling cascade, would react to the amount of insulin in the body and carry this information into the brain where it would cause the observed change. Protein kinase A has been proposed as a mediator between insulin and observed phosphorylation of proteins. The experiment showed that fasting causes increased pKa binding signal. Also, when pKa was inhibited, the phosphorylation of SRGAP3 was decreased, further supporting the idea. pKa was found to form a complex with SRGAP3 in wave 1 to form SRGAP3 wave 1 pKa complex that may facilitate phosphorylation of SRGAP3. Phosphorylation of SRGAP3 further led to increase in its inhibitory effect on RAC1. RAC is a protein that plays a role in cell migration. When it is inhibited, the cell migration controlled by RAC1 was decreased. What happens when SRGAP3 is lost or inactive? Individuals affected by such disorder may experience mental retardation. In rats, this negatively affected memory and learning. Phosphorylated MARCs phosphorylate MAP4, MAP2C, and tau proteins that in turn detach from microtubules and cause dynamic instability of microtubules. High expression of MARC4 and MARC4 tau interactions were detected in brains of patients suffering from Alzheimer's disease. The study has shown that MARCs may be dysregulated due to insulin imbalance. The newly discovered pathway could provide a link between neurodegeneration and diabetes.
So I'm interested in the hypothesis that insulin in some way affects the blood-brain barrier in such a way as that then that sends further signals into the brain that perhaps alter dopamine levels, perhaps inhibit dopamine signaling. And the reason that I favour that as a possible hypothesis is that many of the sites that we've identified to be altered, we know are sites that can be phosphorylated by protein kinase A, cyclic AMP dependent protein kinase, and that is a kinase that's regulated by dopamine.
To sum up, finding out the exact mechanism in which insulin can affect the inner brain machinery and other organs could spur a novel method of prevention, diagnosis, and treatment of the several different manifestations of diabetes. Glucose regulation is only a component of this concept.